All right. So this next talk, thanks, Geraldine. This next talk will be really an intro, as Geraldine mentioned. And a lot of you might already know a lot of this material. That's OK. You can space out for the next half an hour if it's not, none of it is novel. But really, you'll see a lot of terminology, a lot of concepts in the next couple of modules. And we want to make sure you know what we're talking about. So I'm going to give you a very fast, furiously paced overview of next-generation sequencing data from start to finish. All right, let's see if I can use the pointer. Great. All right, so this is the flow today. We'll talk generally about next-generation sequencing, NGS's next-generation sequencing technologies. We'll talk about some experimental designs for the projects you might want to have if you ever do a sequencing project through NGS. Some of the data formats. Oh, yep. Question already. That was good.
Sorry, just wanted to know, would these slides be available?
Will the slides be available? Yes. Yes, they will. I guess different speakers will have different policies. I'm happy for people to jump in with questions at any point. If we're running behind, just Geraldine will let me know and we'll stop questions then. Is that OK?
Yes, but the people that have questions, they need to use the mic. Otherwise, they won't be recorded, or you can repeat the question.
Got it. Questionnaires should use the mic, or they won't be recorded. Or I can see it loudly. OK, then we'll talk about the data formats quickly. I'll show you an example of what kind of files we use. And then a quick overview of the data processing, which Ami will then go into more details about. OK, so next-generation sequencing technologies. So what does NGS do? Really, the illustration here is you have your DNA. And the concept here is as follows. You take your DNA and you fragment. You turn it into lots of fragments, lots of little windows or reads into the genome. So if you notice here, for example, this is one little piece of the DNA. We don't know where it's from, somewhere on one of the chromosomes. And it's kind of knocked into these smaller fragments, these smaller pieces. These fragments can be of different sizes. But generally, today, we're talking about reads, for example, with Illumina sequencing of, let's say, 100 base pairs. So you have a 3 billion base pair genome. And this is one little snippet, one little window into the piece of DNA. And those things get amplified, and we'll move down here, and massively parallelized, sequenced on the machines. So you have, basically what you have is you have a file that contains the base pairs that correspond to all these little windows here. So you go from this big piece of DNA to lots of little pieces. And these little pieces don't overlap. So at any given position in the genome, you might have several reads, several little windows that overlap and show you the base of that composition of that particular position. OK? And then by the end, you get the sequence here of the DNA. So that's really the concept here. OK? And so what we want to actually explain to you with this slide is that as the decades move on, the sequencing machines get better and better, and they produce more and more output. So not only do you have to be accurate, but your tools that process data, or NGS data, have to be really, really fast and efficient. At this point, we're dealing with terabytes of data for lots of projects. And I don't mean like 1 terabyte. We're talking about hundreds of terabytes of data for particular projects. If the GATK can't process this data, it'll just fall apart, and you won't be able to actually run your project. So it's a lot of work not to just get accurate tools, but get to tools that run on all this data. So this is quick terminology. Those in the sequencing department here can probably explain this even better than I can. But I just want to give you, again, the high-level terminology and concepts so you know what we're talking about later on. And this will be very important later on for one of Ryan's talks, where he talks about calibration of data. So what you have really, remember we talked about those fragments a couple of slides ago? This is a little piece of DNA. What happens basically is these fragments are all part of a library. And these libraries, really, you'd have one library per sample. And a sample could have more than one library. But basically, a library is a sample's worth of fragments. And that's the way to see this thing. OK? Then when you look at the sequencing machine, this is kind of what goes into an Illumina machine. You have a flow cell that gets put in, and this gets sequenced. And on the flow cell, you have different lanes of data. Now, on any given lane, you can have multiple samples. The way you pull them out later is you give each sample a unique barcode, and then they can later be sorted out afterwards. But why is this important? Well, sometimes, let's say there's a problem with a whole flow cell where there's an issue, a data processing or error or issue. Well, you might have a covariative error that applies to the whole flow cell, like someone accidentally jars the machine and there's a misincorporation of a base. And that actually happened at the Sanger recently, where they turned off the machine and then turned it back on, and the machine forgot that it had actually incorporated a base and reincorporated it again. So every single read that got sequenced on this particular flow cell had an extra base at the exact same cycle. And so this is the type of error that we'll try to capture in the GATK before we look for mutation variant detection downstream. Also, you can have particular errors that just belong to a lane. Let's say someone sneezes on one of the lanes by accident. That's also possible. Anything that can go wrong, and you really want to divide up the data into the smallest unit possible, so you can really find errors that are unique to those little smaller pieces. Okay. Once you have tons, we're talking about millions and potentially billions of little fragments, those reads of data, we call them the reads, you then need to align them to the genome. You need to find their exact locations, or we'll say approximate locations in the genome, so that you can use them to assess whether there's a mutation at that position. And this alignment process is very computationally intensive. Mauricio, I think, will talk about that a little bit later. So I won't go into too much detail, but this is hugely important to understand that those pieces actually need to be mapped back to their original locations on the genome, as if you can imagine the genome, this two-dimensional object in space. And this is what an IGV screenshot looks like of that aligned data. So we will be showing you a lot of IGV, integrative genomic viewer screenshots. It's developed down the hall from us by someone named Jim Robinson and colleagues. And it's a fantastic, fantastic tool for visualizing next-generation sequencing data. So each one of these, wrong button. Each one of these gray bars represents one read of data aligned to the genome. So this is your reference genome here. I can't see where we are exactly, but we're somewhere in chromosome. I can't read it at this point. Somewhere on one of the chromosomes. And this is what the DNA actually looks like here. And these reads are aligned to this position in the genome. Now, you notice they're all gray. There's no color here. That means that the bases at every one of these positions matches exactly the reference base. Right? But then you have this position here where you see some mismatches. There's some T's where the reference base is a C. Well, this is what a perfectly looking heterozygous SNP looks like. A single nucleotide polymorphism, where the reference is a C, but this sample happens to have one of their chromosomes represents a C, the other has a T allele. And this is what it looks like. So you see mismatches to the reference will show up in color. And the brighter the color, the higher quality or confidence we have in those bases, which we'll discuss in a second. Another thing to notice is that it's a little bit blurry, but some of these reads here all have directionality. They have arrows. This is blunt here. It starts here and moves in this direction. And this arrow moves in this direction. So this was sampled from the forward strand, and this is from the reverse strand of the DNA. Okay, and a lot of these, what this here represents is purple, is paired sequencing. So sometimes you take these fragments, and you'll sequence them from both ends, and they can overlap. That's what you see here. You see this represents one fragment, and it was sequenced from this end here, and then also from that end here, and they overlap. And the GATK, or at least the unified genotyper, takes into consideration that these reads will overlap, and they really represent the same fragment. Okay, is that clear? This is very important to understand going forward, because you'll see a lot of pictures of these IGV screenshots. Okay. All right, so I'll talk about some experimental designs. If you aren't going to be running these designs in a lab, this might just be a nice overview, but you don't really have to focus here. But it will help for downstream, when people talk about how to run their tools on which data sets, this will be kind of helpful. All right, so the main focus here is that given a fixed budget, there's really two major designs. You can have few samples at high coverage. That means at any given genomic position, you have many reads that overlap it. Okay, so for example here, you'll talk about a single sample with 30x coverage. That means there's 30 reads on average at any given position in the genome. Or you can have a lot of samples with fewer coverage. So something like the 1,000 Genomes Project has 2,500 samples, each at 4x coverage. There's only 4 reads at any position in the genome for that particular sample. And one thing you notice here is on the deep single sample project type, you'll see that you have a good chance of finding the variants, the true mutations. But you only find the mutations for that particular sample. If you're interested, in a disease study, for example, you might want to try to use lots of cases and controls together. Here, you might miss a particular variant on a sample. Again, if you only have 4 reads at any genomic position, and a sample is heterozygous, well, on average, they'll have 2 reads with a variant, right? That'll show that allele, the alternate allele. But just by sampling and normalization, it could be that there are no reads or only 1 read, and you might miss that variant on a particular sample. But you're more likely to find more variants in the whole population. Okay? So, for example, here, if you take the we call high-pass sequencing, this is the deep coverage. This is the single sample or few sample, lots of sequencing. Again, you're targeting the whole genome. So this is 3 gigabases, 3 billion bases. And you have usually about 30x coverage. We really recommend 30x coverage, 30 reads per base, to really be accurate in genotyping and finding the actual genotypes of your samples. Whereas in the low coverage cases, again, we'll talk about that in a second, but in the end, you'll find almost all the variation for a sample. Okay? Every single one of those variants you should be able to find, assuming you've done your mutation calling accurately. Low-pass design, again, is a little bit different. Again, you're still targeting the whole genome, but your coverage is much lower, and you won't find all the variants in the sample necessarily. So whereas before we were finding 3 to 5 million variants per sample, now per sample it's really capped at 3 million maximum. Okay? But you'll find a lot more variation in the population-wide. Okay? And there's another type of experimental design, and that's the one we really see the most at the BroadE, because we deal with a lot of medical genetics projects, and that is the exome capture project. And that's really, instead of being 3 billion bases, we're talking about 32 million bases. It's really just trying to cover genes and surrounding regions. And the coverage there is very variable, but we are aiming is for more than 80% of your targets that you're trying to capture of the genes to be at greater than 20x coverage. And there's usually, for most of the targets that you're trying to capture there, usually see something like 100x, which is good. Here, again, you're not finding 3 million variants per sample. You're seeing something closer to 20,000. It's a little bit on the high end. But hopefully, these are variants of genetic interest because they fall in the exons. Someone a question? Yep.
How about for genomes that have high repeat content? What's the
I missed that. High what content?
repeat content?
Repeat content. Well, the human genome also has a high repeat content too. I suppose this is probably not the right module to discuss it. Maybe you should ask that. Can I ask you to? You're talking about for finding variants, right? So I'm going to ask you to repeat that question for Ryan and Guillermo when they talk about the actual SNP calling later on. Is that okay? Okay, but it's hard. I mean, in the end, if you're talking about finding SNPs and repeats, it's non-trivial. We're working now on finding STRs, short tandem repeats, and we're trying to do a good job there. But more details later on, okay? All right, data format. So I want to show you some files, show you the files you'll need to know. And there's a lot to cover here. So you don't have to know all the details. If I say things that are a bit fuzzy, don't worry about it unless it's completely unclear. I just want you to have seen it first before we talk about it. All right, so the overall flow looks like this. You have your big sequence machine, and from the raw data, there's something called a base caller, which is the machine basically, it's the machine software that says, I saw an A at this position, then I saw a C, and I saw a T on this read. And it basically gives you these strings, these lists of characters. And they're put into FASTQ files over here. The FASTQ file represents the raw output data of the sequencing machine. Then we have that alignment or assembly step where you take those reads and map them to their places in the genome, and the SAM or BAM format represents those. These are aligned reads to the genome. And finally, after the variant calling step, you have a VCF file. The VCF is the variant call format and represents the mutations, the variants inside your sample data. Okay, so we'll go over each one of these in turn. So the FASTQ file looks like this. Again, this represents the raw output of the sequencing machine. And the important piece is there's four lines here. Okay, the first one is a unique identifier for that particular read. Every read should have its own identifier. Then the actual base content of that read. So this is whatever 20 base pair read, and this is what the machine saw. Skip the third line. This here actually represents a confidence, and I'll explain this in a second, a confidence in each one of those bases. So each one of those bases has another character here that represents the confidence. Now this is just ASCII and represents a number in ASCII format. And what this number is really the machine is saying, I am X confident in this call. So I said, I saw a C here, and I'm 99.9% confident it really is at C. Or it might say, I saw a C here in this position, and I have like 75% confidence in it. And that's very important for downstream SNP calling. You see a lot of bases at a given position that'll have very low confidence, well, it's probably not a real SNP. If you see a lot of bases at that position that all have you're very highly confident non-reference, then you're much more likely to make a SNP call there. Okay? So what I talked about here with these confidences, these are Phred scaled. And I've given this presentation once before, and people had no idea what Phred scaling was. So I'm going to just jump in here and explain to you what Phred scale is. Almost every presentation we'll talk about later on talks about Phred scaling, Q-scores. So I just want you to understand what it is. So this is the actual mathematics about it. It's a negative 10 log 10 of the error rate. Okay, you're 100% minus your confidence. What I mean is like this. If you're 90% confident in a particular base or a SNP or anything, then you say you have a 10% error rate, right? And that's considered a Q10. It's exponential. If you're 99% confident in a 1% error rate, it's a Q20. So Q30 is 99.9. Q40 is 99.99, et cetera, et cetera. So when you're getting to things like Q50 already, you're 99.999% confident. That's probably more confident that you can actually be with next generation sequencing. So if you start to see Q scores, for example, in your bases, so that you're saying the machine is Q50 confident in its base, you just can't really believe that at this point. Now, I'll say downstream, Ryan and Guillermo will talk about our SNP calls, and there'll be a confidence there, which is also Phred scaled, and will be like Q a million. Those are not accurately represented, we'll say. Okay, so don't be afraid if you see those. And we talked about here, this BAM here, we have these numbers. They're ASCII 33 plus the Q score. So if you see that, it's because in ASCII, the representation, the first 32 bases, the first 32 numbers don't represent visible characters. So that's why in the BAM format, they add 33 to the number. Okay, these are just details, so you know if you ever look at a BAM file and you see these in IGV screenshots when you overlap, you're not afraid when you see these Q scores. Okay. BAM format, this is when you store the aligned sequence data. And so what happens here is you have lots of different sequencing companies, and they all had originally their own data formats. And it got to be a nightmare to actually try to process all of them. PacBio would have their own format, and Solid would have their own, and 454, and Illumina. So we basically all converged on one particular format, the BAM format, or the SAM format. BAM is binary SAM. And it basically looks like this. You remember that unique identifier for a read? This is a given read in the file. This unique identifier is converted over here. Right, so we still say here. This is the chromosome 1 and position of this first base where it's aligned. So you have this read, and it aligned to chromosome 1 in this position here. And this here is called the cigar string. This kind of dictates how that read is aligned to the genome. So for example, if it's straight alignment, then it might be, let's say it's a 100 base pair read, it'll say 100M. But there could be insertions or deletions in that read relative to the reference. So those are also mentioned in this tag. You'll see, for example, 20M, I6, and then 74M, which means that the first 20 bases are aligned as is. The next 6 bases represent an insertion and don't actually align to the reference. And then the next 74 bases align as is to the reference. Okay? Again, just so you see later on, if you see cigar strings, you're not thrown for a loop. This is again, the DNA sequence, the actual bases. These are the quality scores we mentioned before, those Phred scale quality scores, the confidences in each one of these bases. And you have other tags also, which we'll discuss in a second. Okay, this is the header of the BAM file. So before you actually have that sequencing data I mentioned, you have the header. Now, why are we showing you the header? It's because if you don't have well-formed headers, the GATK will barf on your file. We'll say, go back, put the headers back in, and start again. There are lots of things to point out here. The most important one is you have these things called regroups. Remember those libraries we talked about in the flow cells? Well, each one of these regroups basically represents one lane of data for a particular sample. So again, you might have 10 samples in any given lane of data, and each one of those samples would have its own regroup. So you'd have 10 regroups representing that lane. And this is a kind of a rough estimate, but this is really, again, high level. And so why is that important? Because the regroup tells you, first of all, the sample name. So when we're calling SNPs later on, we're using this information to divide up the reads into the appropriate sample bins. And also, Ryan will show you later, the sequencing machines make mistakes. And we kind of also want to find these, which errors are associated with which read groups. Each read group has a different, really, covariates of error, different extent to which they are errorful also. Okay? Again, when you see these, I have the links here. So later on, when you just get these presentations back at home to peruse over coffee, you can go back and look at more details here. And finally, the VCF format. This is the format that represents the mutations inside your data. Again, the header here is not quite as important as the BAM header, but we're not going to go over it. We're just going to go over the last header line. I should also say that because of formatting issues, from this line and forward, each 2 lines is really one long line that I couldn't fit in. It was just going to wrap over. So this is all one line here. So this is basically a tabular delimited file. So you have the chromosome of the mutation, position, some unique identifier, usually a DB SNP ID for human data, the reference allele, the alternate allele. And I'll stop there and I'll show you an example. So this first mutation occurs on chromosome 20 at position 14370. It's in dbSNP, and this is the dbSNP ID. So this one here has no dbSNP ID, means it's not a dbSNP. Okay. The reference base is a G, and the alternate allele is an A. So this is a SNP, G to A SNP. Okay? Now we'll go to the next few fields. Quality is at Phred scale quality. So we're approximately at Q29 confident. So again, Q30 is 99.9% confident. So we're 99.8% confident in this call. Pass means, so the filter field is it good or not? Again, Ryan, later on, tomorrow, I think, will talk about filtering calls. And so you will learn about, you'll see other things here that are not pass. But for now, this is a passing record, and it's a good real variant. And then you have the info. Info is some like metadata about their position. Not per sample, but overall the position. So DP, for example, is the depth. So there were 14 reads at this position. The AF is the allele frequency. So this is the allele frequency says, of all the samples I saw, half of them, 0.5 of them, had the A allele over the G. Okay? And there are other ones, too. Then you have the sample data, the format, and then these are our samples, NA0001, 2, and 3. These are 3 samples, and this tells me how to read the following columns. So GT, GQ, and DP are 3 kind of metadata pieces per sample. Every sample will have its own metadata. And this is important, by the way. You really need to see this because later on in presentations, we will show you VCF records that you'll want to understand. So what this is saying here is that the genotype for the first sample, NA001, is 0, 0. That means there's 2 chromosomes because we're diploid in humans. And 0 represents the first allele, G. So this sample is GG. She is the reference. She's homozygous reference. Okay? GQ is some sort of quality, which we don't discuss now. And the DP is also the depth. So she has 1 read in the sample, and she's homozygous reference. So it's not a very confident call. You only have 1 read that overlapped that position for that sample, and we're saying, well, that was a reference read. We just think she's reference. We don't see any evidence for a SNP. So not a very confident genotype for this sample. Now the next sample, NA0002, is 1, 0. That means 1 represents this base here. Okay, so the genotype here is heterozygous. There's a G and A with 8 reads. So we're a lot more confident, potentially. And then this sample here is 1, 1, AA. So this sample is homozygous variant. Now, you might get thrown for a loop for this record down below. There's a GT. You can represent more than 1 altered allele in any record. Right? So you'll see this sample here is 2, 1. This person has a GT genotype. Reference is A. So you're a heterozygous variant. And this is a TT. Right? 2 is the T. 1 would be G, and 0 is A. And no one here is actually reference. Okay? So that's a lot of detail about the VCF, but you'll need to know that for modules downstream. I know that because I wrote one of them, so I just want to give you that. Okay, and finally, the data processing overview. We talked about conceptually, you have DNA, goes through the sequencing machine, and the sequencing companies would love you to believe that right out of the sequencing machine, you have this big VCF file with all the variants, mutations in your sample or set of samples, from which you can go do medical or population genetic studies. And it's nice and simple, and you should buy their machines. But it turns out this step here is actually a little bit more complex. And it's because the data is not so easy to work with. You remember that first IGV screenshot we showed you back with that nice, beautiful SNP? Well, we kind of had to doctor it a bit to make it look so nice. This is what a usual screenshot looks like. You see all these red and blue dots everywhere? Again, these are mismatches. It's kind of blown. We step back a little bit so we can put it all on one screen. But how much of the stuff is actually error? You see up here, there's a depth track, and there's a lot of color here. So a lot of these could be real SNPs. A lot of them could be not real SNPs. How much of this stuff is actually error, and how much of it represents real variation? Well, that's what the GATK does. We try to weed out the error and find the true variance from your samples. Okay? And this is what this pipeline is. So Geraldine mentioned this before, and again, Ami will give you more detail here. But it's a non-trivial process here. First, you have data processing. You take the raw data coming out of the machine, and you have pre-processing steps to even get it into a format ready to do variant detection and mutation detection. Then I think we do this tomorrow, Geraldine, right? We talk about the mutation and variant detection tomorrow. And you can detect SNPs, indels. Structural variation will be this afternoon in Bob's session. And then finally, there's kind of like the post-processing step, which is integration and filtering, things like that. And only then, after you've done all these steps through the GATK mostly, do you have analysis-ready variants. So this is what today's and tomorrow's talks will be all about. I should also point out with the GATK, there's kind of light and full versions. For academic users, it's not really an issue. Everyone gets the full version. But there's new functionality that's been added where we're really kind of trailblazing here and leading the pack as far as better, in particular, better indel and larger variant substitution calling. And also being with the ability to call really larger sets of samples together, larger batches. I'm not going to give you details here because I think Ami will and other people will too. Finally, I should point out that we don't write all the tools to process next generation sequencing. In particular, I like to point out the Picard set of tools and Samtools, which we use also. They have a lot of nifty tricks and tools they have available for them. So you should check them out if you're really going to do any processing of sequencing data. And that's it. Are there any questions? Good. There's just an overview. No? Good. Oh, question. We have 5 minutes for questions if anyone wants to ask anything. Good. I hope it's because it was clear, not because you were totally.
Actually.
Sure. Can you stand up to the microphone? Is that OK? I'm sorry.
The VCF format, do you think you could go back to it? I had a couple of questions about it. So these three NA0001, 2, and 3, what do those three things represent?
These are the sample names. It could be Eric, Geraldine, and Ryan. Usually they're anonymized. I don't know how that word is, but because it's sample data. So this is just the column headings here, and this is the column representing this sample, that sample, and the third sample. And so it would match up if I could spread it out all in one line. So NA001 here has a reference genotype, 00. Here she has the GT genotype. Here it's reference, TT. And so I should point out here on this one, there's no alternate allele. This is a reference site where there is no alternate allele. And you can represent that in VCF too.
Right. OK, thanks.
Good, no problem. These are indels, you should also say. So the GT, CT is reference, and one alternate allele is a G. So you've done a deletion of 3 bases. GT, CT to G. So you've lost these 3 bases, though. Sure.
So I saw the 0 for the second sample. There's 0. Yeah, down.
This sample.
2, 1, 2, 0. I guess 0 steps one.
Yeah, I don't know.
Genotype call.
Whoever put this particular example in chose poorly for the depth. I think they were just putting numbers in to make it as an example. So it's a good question.
Okay.
We should probably put a real life. It was hard enough to get this formatted right. I'm not going to spend more time to. I should update those numbers. It's a good point. Thank you.
So does it mean that GATK is now no longer limited to just diploid organisms?
Yeah, I don't think so. There are tools in the GATK that can handle SNP calling in non-diploid organisms. But we're not going to cover that in these modules. I think this is I don't know.
Not specifically.
Not specifically in these modules. This is more. That's definitely that falls under the heading of advanced functionality for the GATK. But yes, we are able to call variants in non-diploid organisms now. Okay.